I would like to thank everybody for being here this morning and for the organizers inviting me. I really appreciate the chance to talk. I have nothing to disclose financially here, and I just want to thank all of the people and the foundations that have supported this research: the National Institutes of Health, the American Brain Tumor Society, Southeastern Brain Tumor Foundation, and the American Cancer Society. So I'm not going to try and lecture this audience on glioblastoma. Obviously, there's many more informed people in the audience. But what I would like to tell you is another perspective of how we're trying to understand this disease. Sorry. So I'm interested in glioblastoma, which is the most primary and malignant tumor in the central nervous system. These patients suffer a poor prognosis very short after they're diagnosed. And it's because it is such an aggressive tumor that diffusively infiltrates. What we know about these tumors is if you look at different sections of the same tumor, or even tumors between patients, they don't look at all alike. They're very heterogeneous in nature. And so what I should mention is that traditionally, a pathologist would look at this and classify this tumor based on the cell of origin that it was likely to come from, and then how bad the disease was. What we as basic scientists are trying to do is to provide more information to you. And so we're considering a molecular-based classification that in fact might be assisted. So considering this molecular-based approach, the NCI supported an endeavor to do mass genome sequencing, proteome, everything that you could tell about these tumors. And here's what we came up with. Essentially, in GBM, despite how diverse they are, there's essentially 3 altered signaling pathways. So in about 80% of them, they will have disruptions right at the level of receptor tyrosine kinases. And what that's doing is it's sending pro-growth signals in. In about 87%, they've essentially lost p53 tumor suppression, so we lose the ability to induce apoptosis in these cells. And then in about, I think it's 80% or so, we've lost the ability to properly regulate the cell cycle. So this was particularly informative because it told us that essentially all GBMs would have disruptions in these 3 pathways. And what it would allow us to do is to focus our research and therapeutic endeavors on these targets. And so you can see that a lot of novel therapeutics have come out for GBM, and it's been based on a lot of the information generated by the TCGA. For example, inhibitors that would target some of the receptor tyrosine kinases, or the downstream pathways. What I should mention is essentially a lot of that focused on regions that code for proteins. Now, that part of the DNA only counts for 2%. The other 98% of your DNA, a lot of people thought was just junk. It's not junk. They're non-coding regions that can affect everything else. And what I'm here to tell you about is that that can actually have an impact on cancer as well. So some of the groundbreaking work came out of Carlo Croce's lab at Ohio State, and he had been working on CML. And what he had said was, there were a lot of deletions in one area of chromosome 13. And when he went and tried to map which genes were likely responsible, he couldn't find that there was a correlation between them, between the open reading frames of the protein coding regions and the disease. But in fact, it was 2 microRNAs that were essentially conferring susceptibility to this disease. So I'd like to introduce microRNAs. And essentially what they are is they're just like an mRNA in the sense that they have a gene located in the genome. They're transcribed, and in their full mature form, they're quite small, only about 20 to 20 nucleotides long. And what they do is they get incorporated into a complex, and then they regulate gene expression. So this is just a diagram showing you that essentially a microRNA is encoded by the DNA. It is transcribed in the nucleus and then processed to reveal a smaller form that then is put out to the cytoplasm, where it undergoes a second cleavage. In the cytoplasm, we essentially take this small piece of RNA and incorporate it into a protein complex. And what that does is it serves like a heat-seeking missile. that microRNA will look for mRNAs that have similarities, and it will bind to it and essentially prevent the gene expression. So the important thing here is that one microRNA can regulate the expression of hundreds of mRNAs. Consequently, if you lose a microRNA, you lose the ability to regulate hundreds of genes. Now, other people have been interested in microRNAs and GBM, and they've already implicated a number. So miR-21 is probably the most potent, and we know that it can target P10, one of the most prominent targets in GBM. But the problem is, where we were seeing percentages like 87% in the open coding regions, we only see that collectively these microRNAs account for about 5% of GBMs. So we haven't found the one yet. Now my interest is in the NF-kappa B pathway, and this is a pathway that mediates inflammation. And I think a lot of you can appreciate that inflammation correlates very well with the onset of tumors or cancer tumorigenesis. So this is the pathway. NF-kappa B is a dimer of P50 and P65, and it's kept in the cytoplasm and in an inhibited state. And that's because you don't want to have an inappropriate inflammatory response. But should you want to turn on inflammation, the cell will respond to something like TNF, which binds to its receptor, trimerizes, and then recruits additional proteins. Now there are many. I'm only going to tell you that you just need to worry about TRAD. So TRAD is the conduit between signals outside the cell and signals inside the cell. And what it does is it activates the cascade that causes NF-kappa B to turn on. When NF-kappa B is turned on, it turns on hundreds of genes. And collectively, those genes mediate things like proliferation, angiogenesis, adhesion, and invasion, preventing the cell from dying. And so you can imagine that if a tumor could somehow hijack this pathway, it would be very advantageous to it. Because the tumor would be able to proliferate, prevent cell death, become migratory, and encourage new blood growth. So we and others have already shown that if you look at a number of cancers, and this is examples of GBM, that the levels of NF-kappa B, total and activated, are much higher in gliomas, as shown here, than in the control samples with a non-neoplastic tissue. And when we associate that with the genes they can induce, these collectively, which are all involved in preventing cell death, what we know is that NF-kappa B is expressed and elevated in these tumors, and is causing its target genes to be elevated as well. And that's a problem because, again, it's mediating things like protection from cell death, growth, and so on. So we thought, let's look for a microRNA that could potentially regulate NF-kappa B or be regulated by NF-kappa B. And what we identified was microRNA 31. I'll refer to it as miR-31. So to ask if it has relevance in cancers, we went back to the TCGA, and because they had successfully evaluated a number of cancers, we could ask how often was this microRNA dysregulated in cancer. And what you're seeing are instances where both copies of the gene is lost, as indicated by red, or sometimes where the gene is amplified, as indicated in green, or both. But in those instances where we have both, the deletions always outnumber the amplifications. And I hope what you can appreciate is the largest bar essentially belongs to glioblastoma. And what it says to us here is that 37% of all GBMs will lose both copies of this microRNA. And in fact, we see that 7% of the lower grade gliomas will as well. If we extend that to how many tumors will lose one or both, the number jumps up to 70%. Now, it's not that surprising, in fact, because it sits at a locus in the genome where we've known a lot of information about it. So this is 9p21.3, and we've known for some time that this is the area of DNA that you're most likely to lose in GBM. And what we know is to the left of microRNA 31, which is encoded by this gene, are two genes which encode 3 tumor suppressors. So essentially, when they lose this, they become very vulnerable to becoming tumorigenic. And then to the right is the interferon alpha locus that we've known about for some time. Now, we could say that the loss of miR-31 is simply because you're losing this, but what we find is that, in fact, either the tumor will keep this entire area intact, that's essentially 21% of the tumors will not lose any of the genes, or 71 will lose one or more copies of every single gene in this area. So So it's never a focal deletion of just these two. In fact, it's a deletion that takes out all of this. So we think that the loss of miR31 has a contribution to the tumor formation as well. I'm going to try and keep it away from too much hardcore science, but again, what we wanted to know is, could this be regulated by NF-kappa B? And if so, what was its purpose? So essentially what this is telling you is, if we use TNF, which would activate NF-kappa B, and if NF-kappa B is regulating miR31, we should see a positive correlation. And in fact, we do. When we add TNF, after several days, we can see that there's a rise in the levels of miR31. And this confirms that it was a direct target. Now, what I need to mention is, if you have a microRNA, it's going to target an mRNA, and there will be an inverse correlation. So if NF-kappa B is turning on a microRNA, whatever target it's going after should be shut down. And so what we were asking is, who would it be targeting? And what we identified was that TRAD, the upstream regulator, was a target of miR31. So remember, it's very important to establish proper control over the inflammatory pathway. You want to keep it off. When you activate it, it should be necessary, and then you want to restore it to an off position when appropriate. So what we think is that NF-kappa B induces the expression of miR31 so that it can feed back and turn off the pathway when necessary. And so you can imagine in the tumor, if they've lost miR31, they don't have an ability to come back and turn the pathway off once it's on. So we could confirm, if we looked at various glioma cell lines, that where they had miR31, there were much lower levels of TRAD than in those tumors where they did not have miR31, and the levels of TRAD were much higher. So what you could say is, if we have high levels of this protein, then everything downstream of it should be high as well. So here's our model. Essentially, NF-kappa B is the pro-inflammatory molecule that should be carefully regulated. It can be turned on, but essentially to do that, you have to work your TRAD. To ensure that NF-kappa B is properly regulated, it will turn on two things. I-kappa B, which will come back to inhibit it, or miR31, which will come back and inhibit it further upstream. And here's the problem. Marcus Burdell at UAB had shown that 20% of tumors will lose one copy of NF-kappa B, and this was segregating to the non-classical GBMs. So what we know is that in those tumors they have high NF-kappa B activity, and we could give it a genetic explanation. The problem was, what about the other 80% of GBM, where they had both copies of IgA, or I-kappa B alpha? What we found was the relationship was mutually exclusive. So either you lost a copy of NF-kappa B, or you lost a copy of miR31. But the consequence was the same, that in both tumors you got an NF-kappa B that was too high, and now it was promoting angiogenesis, self-proliferation, and resistance to death. The interesting thing was that this segregated to a discrete site of GBM. So the important thing is to ask, well, what does miR31 mean for the tumor? So what we've done is gone with patient-derived GBM xenografts. So these are tumors that are removed from the patient and immediately disaggregated and then passaged in the flanks of mice. And this is important because they experience a better in vivo microenvironment. So these are those tumors, and we know that these have microRNA 31. And what we can do is leave those levels present, or we can antagonize them with an antagonist from miR31. So what you're seeing on the right flank are those tumor samples that contain miR31. And what you're seeing on the left are the same tumor samples, but where we've removed miR31. I hope you can appreciate that they grow much larger in a much shorter amount of time. So we think that miR31 is there, and that part of its response is to keep NF-kappa B in check so that the tumors don't grow so big. And that essentially, if you remove it, part of its contributions is making NF-kappa B too active. The other thing that we noticed is if you've lost miR31, and these are those same tumors, we're just looking at the pathology. These are the tumors that have miR31. There are a lot of cells, obviously, but when we've taken away miR31, what we see is that we've also increased the angiogenesis. And again, that's something that we would expect to correlate with high activation of NF-kappa B. We also see that there are changes in NF-kappa B expression and the target genes. And most importantly, when we translated this to intracranial models, we can see a difference. So again, what we do is we take these glioma cells, and the only difference between the two is whether or not they have miR-31. And we implant these into the brain of nude mice. And what you can see here by the blue line, these mice received glioma cells that did not have miR-31. So all of the mice were characteristically dead by 30 days, but when we gave them miR-31, we noticed the mice were living a much longer time. And I should mention, one of the mice was killed at these same time points because people believed that they weren't forming the tumors. And they weren't. And I should mention that this one, in fact, never did. So this is quite exciting because it suggests that miR-31 could be used to inhibit the growth of a tumor. Now, why is that particularly exciting? Well, microRNAs are being considered in clinical trials. They're so small and they can readily pass between membranes that they are being considered for use in treating tumors. I should mention, if we look at human patient data, this is from the TCGA, and what you're looking at are instances of patients where they contain both copies of miR-31 and patients where they've lost both copies. And although it's very slight, it appears that the presence of miR-31 may confer a better overall survival for those patients. So again, the model is that signaling that activates NF-kappa B should be normally kept in check by factors such as I-kappa B alpha and miR-31. But again, we have a problem because we know that I-kappa B alpha is deleted, as is miR-31. What you end up getting is NF-kappa B that is unregulated, and it's going to contribute to tumor growth. Now, I would be naive to say that the only way that miR-31 is working is through NF-kappa B. So what I want to do is to tell you about the population of cells that we are thinking are the most problematic in treating these tumors. So you might have heard these termed cancer stem cells, or brain tumor initiating cells, or cancer propagating. What I want to do is to introduce the concept that the tumor itself is not homogeneous, that it exists of different types of cells. The majority of the cells exist as a more differentiated and susceptible population, susceptible to the traditional chemotherapies and radiation. But it's the minor fraction that's less differentiated, and it is that population which, left untreated, will essentially reconstitute the tumor. Now, we know that with regards to normal stem cells, the things needed to keep those immature, less differentiated cells in that state include things like NF-kappa B, stem cell factors, and polyclonal repressor complexes. So what we know from work from the Yamanaka's group said that if you had a fully differentiated cell and you could somehow activate NF-kappa B, reintroduce the stem cell factors, and reacquire the polyclonal repressor complexes, you could actually reprogram that cell and make it into anything you wanted. So I'm not saying that cancer is doing that, but maybe it's got some version of that where it can take a differentiated cell and reprogram it, or at least take a less differentiated cell and maintain it. So it can do this because the stem cell factors will turn on genes that are needed to maintain this pluripotent state. And they will turn off genes that are needed to promote differentiation. And they do that by working with the polyclonal repressor complex. It essentially modifies the DNA so that it's no longer accessible. What we know is that many of these things, NF-kappa B, the stem cell factors, and the polyclonal repressor complexes, have already been identified as targets in GBM. We know that they're overexpressed and they're amplified, and that they're critical for maintaining that stem cell population. So what we're going to ask is, could miR-31 have a role in determining whether these cells remain undifferentiated or differentiated? What we're going to use are neural precursors as models of undifferentiated cells, or astrocytes as models of differentiated cells. So what you're looking at here are the levels of genes associated with certain things. So OCT4 is associated with stem cell machinery. We would expect to find it in cells that are less differentiated. And in fact, in the neural precursors, that's where we find it. But in a cell that's differentiated, such as the astrocyte, you can see it's almost absent. Now, GFAP begins to show as the neural precursors adopt a more astrocytic lineage and commit to that. So we can see that GFAP is high in the astrocytes, and these are actually astrocytic neural precursor cells, and it's high in them as well. What was interesting is when we looked at miR-31, we found it virtually absent in the neural precursors. So it was interesting to us because it said in an undifferentiated state, we did not have miR-31. Now we can take these neural precursors and force them to differentiate. We can do that by adding FBS, which is just serum, or we can use BOP4. And what we've done is changed the fate of these cells. So these neural precursors will look more like astrocytes, and consequently they'll they'll express GFAP and lose things like nestin and OCT4. So if we force these cells to turn into a differentiated cell, what we also saw was that miR-31 was coming on, and this was quite exciting. So the question was, how is it kept off in those cells, and what is it turned on by, and what is it doing? Now remember, the stem cell factors and the polycomb repressor complexes are great at regulating gene expression. So we asked the question, could they in fact be regulating miR-31? And what we found, to make this very simple, was yes. In a cell where you have high levels of stem cell factors and polycomb repressor complexes, if their job is to keep things that are promoting differentiation off, they do so by binding to the DNA at regions upstream of the gene and modifying the DNA in such a way that it's no longer accessible. So what we're seeing here in the blue boxes are the neural precursor cells that are still pluripotent. They have high levels of the stem cell factors and the effects of the polycomb repressor complexes, and what that does is it keeps it in an off state. But if we force these cells to differentiate, those things exit, and what we can do is now reacquire miR-31 expression. Now, is miR-31 simply turned on as the cell is differentiating, or is it actually mediating the effects of differentiation? So what we've taken are the neural precursor cells, and we've introduced either a control microRNA or miR-31 itself. And what you can see is, if we introduce microRNA 31, the levels of our stem cell factors drop. So that suggests that they're no longer pluripotent. To assess whether they're differentiating, we looked at things like GFAP, and those things were going up. So this was quite exciting because, again, if we can push the immature cancer stem cell population out of their comfort zone, out of this undifferentiated state, and force them to adopt a fate, we might make them more sensitive to the existing chemotherapies and radiation therapy. So this was done with normal murine cells, but we also looked at tumors. And so what you're looking at are those tumors that you saw earlier. Again, these are the tumors that have miR-31. So in the control, what we're looking at are those things that are needed to maintain the stem cell population. So when they have miR-31, they have very low levels of these proteins. But if you take away miR-31, those very factors that help maintain the cell in the undifferentiated state begin to show up. And what we think they're doing is they're helping that population of cells most likely to repropagate the tumor. We think that they're helping them. Now, what happens if you've lost miR-31? Is it enough for a cell to de-differentiate? And we think so. What you're looking at are normal murine astrocytes. The blue is indicating a nucleus, and the green is indicating GFAP, which is a marker of astrocytes. And what we're seeing here are cells that have just received a control microRNA, or cells that have removed the miR-31. And what you can see is that they begin to completely lose GFAP expression, as indicated here and here, and they reacquire nestin and all of the stem cell factors. So it suggests that if a cell were to never have miR-31, it would be maintained in this less differentiated state. And if a differentiated state cell were to lose miR-31, it could potentially revert back to this more immature stem state. And what we know is that the cells don't like to do this. So these are astrocytes that have received control microRNA or miR-31, and what we're measuring is their metabolic health. And they appear to do just fine. It's only when we try to take away the miR-31 that the astrocytes have somewhat of a crisis. And this might just be this kind of event needed to instigate tumorigenesis. So let's ask if we've done that essentially in another tumor model. So again, you're looking at cells that contain miR-31 and cells that do not contain miR-31. We pull those out and we ask about the cancer stem cell population by looking at those functional markers that are needed to maintain it. What you're seeing over here, those cells that have miR-31 have very low markers of the stem cells, and again, the tumors are very small. If, however, we simply remove miR-31, the tumors essentially blow up, and we believe it's because we've reactivated this program that allows that stem cell population to proliferate. Now, why is this important? I think this is important because sometimes, as a Ph.D., we're trying to think, how can we help the MDs? And we have to think outside the box. We can't just keep feeding you the same information if it's not working. And so we can take a lesson from what's happening with prostate cancer, and that is essentially to consider differentiation therapy. So again, when you consider that we're dealing with a tumor that's actually quite heterogeneous, that is, that you have blue cells, which are those cells that are most mature, most differentiated, and then you have cells that are less mature, less differentiated. Those are indicated by yellow and red. Now, what happens when we give them radiation and chemotherapy? Well, the blue cells, the most differentiated, are very susceptible. We can treat them very well. The problem is, those that remain, the yellow and the red, those are the less differentiated ones, and those are the ones that are going to come back and essentially repropagate the tumor. So how do we deal with those? Well, if we could force them out of their comfort zone and essentially force them to differentiate, to become more like the rest of the tumor, we could actually resensitize them to the traditional therapies that you're already using. And because microRNA is so small, and it's currently being considered, and it shouldn't raise inflammatory response, we're quite hopeful that miR-31 might be something that we can use in this respect. Now, I should mention that I work with a team of people who are all fantastic. My technician, Rajni, and then a number of collaborators, Drs. McFarland, Mears, Goodell, Baveri, and Dr. Benvesti. And with that, I'd be happy to answer any questions that you have.